In session 2, we want to be able to count live and dead cells. The first step is to get rid of the old media or the spent media that the cells were sitting in. And there we have just removed the old media. The next step is to ensure that we get rid of any cell debris or any other debris that may be present within the flask. And so we add a little bit of phosphate buffered solution, or PBS, which is essentially like salt water. And so we clean up the flask to make sure there isn't any debris left over during the culture period. And there we toss out the PBS. Our flask is now clean, and it in fact has cells attached to the bottom of the flask. To remove the flask, we use an agent known as trypsin, and trypsin is now being added to the flask, and trypsin will detach the cells from the flask. So we go ahead and incubate it in the incubator for a few minutes and allow the trypsin to do its work to detach the cells completely from the flask. But also we don't want to leave it in there for too long because trypsin also is toxic. So we want to be able to leave it for just the right amount of time so that the cells detach, but not so long as to cause the cells to deteriorate. At this point, we can deactivate the trypsinization process. And the way we do that is simply by adding fresh media to the flask that has the cells and the trypsin in it. The cells are now resuspended in this solution of fresh media and trypsin, and they have hopefully completely all detached from the flask. It may help to look under the microscope at times to make sure that you have detachment of all the flasks, in case you're unsure of whether the cells actually detached or not. At this point, similar to session 1 session, we pipette out the solution, we put it in a centrifuge tube to be centrifuged. Again, disclaimer, for all the numbers in terms of the volumes that were pipetted and so on, please refer to your session protocol to know exactly how much of everything was used. Now that the solution with the cells is in the centrifuge tube, that is ready to be centrifuged. We already showed you these steps in session 1, but effectively you would put it in there with the balance and then get ready to centrifuge. So after we've done centrifuging, we take our centrifuge tube out just like we did in session 1. Again, you can clearly see the pellet at the bottom of the flask. And at this point, we want to then proceed back to the laminar flow hood to prepare our cell counting. So at this point, we want to again remove the media that contains the trypsin, because trypsin is also toxic to the cells. So we remove carefully all the media which contains the trypsin, leaving that pellet behind, as you can see at the bottom of the flask. We then proceed by pipetting some fresh media and placing it into the centrifuge tube and resuspending the cells in fresh media, which is what they like. So we gently pipette the cells up and down within the fresh media. And again, some of these steps you also already saw in session 1, but these have to be repeated here, obviously, for session 2 as well. Okay. So once we feel that the cells have been uniformly distributed into the fresh media, we are at this point ready to seal the cells for the time being and get ready with the next step, which is a key part of session 2, which is counting the cells. Here you have a reagent called trypan blue, which can be used to stain dead cells blue, and live cells don't pick up the stain, so you know which cells are live and which cells are dead. We take roughly, let's say, 10 microliters of trypan blue, and we want to then introduced to the trypan blue, which is in that container there, with an equal volume of the cell suspension. So if you use 10 microliter of trypan blue, you need 10 microliter of cell suspension. So there you can see the cell suspension that is in the micropipette, and that is now being added to the vial that already contains equal volume of trypan blue. That is gently mixed in with the trypan blue. So we have in that vial 10 microliter, let's say, of cell suspension with 10 microliter of trypan blue. And at this point, we take that and insert it into a device known as a hemocytometer that effectively has 4 quadrants, so you can count cells within each quadrant and take an average count of live cells which have no color and dead cells which will show up as blue spots. And in order to enable your viewing of the live and dead cells, the hemocytometer, which looks like effectively like a slide, needs to then be placed under a microscope for viewing. So we have a microscope here, and we set it up, switch it on, we put the slide underneath, under the objectives, we can take a look, and we can set it at the right magnification and focus and field of view and start counting the 4 quadrants. And here's a representative image of what live and dead cells will look like under the trypan blue viability assessment. This concludes session 2.